Hello, welcome to You've Got Potential, a podcast about the work and lives of undergraduate researchers. And today our guest speaker is Thejasvi, and I've known her we've been good friends since middle school, and we followed each other to Rutgers as roommates until the coronavirus, and we've been on top of each other's research paths, and so it was very easy for me to annoy her about getting on this podcast. I didn't feel guilty at all. And so now we have her here. And so if you'd like to introduce yourself and give a brief synopsis about your research.
Sure. So like Monica said, I'm Thejasvi. I'm going to be a senior in the fall at Rutgers. I'm majoring in molecular biology and biochemistry. I'm minoring in religion, so that's pretty different. I'm going down a pre-med path, but I'm super into research. And the research that I'm doing is more of basic science. So we work on C. elegans, and I've always been super interested in embryonic development. So that is what we study, and that's really fun. We study the actin dynamics during different processes, such as ventral enclosure in C. elegans embryos, as well as in corpse engulfment, and watching how those two things are similar and different, and why the same guidance receptors and everything are differentially regulating those things. So yeah.
Cool, thank you. Could you give a brief summary about what a C. elegans is for anyone who is not familiar?
Right. I'll try my best to describe it. C. elegans is a small, it's called a nematode. It's a little tiny worm. You can kind of see them with your bare eye, but you have to use microscopes basically to even look at the animals. You grow them up on little mini Petri plates, and yeah, they're transparent, so that's really good for imaging fluorescent proteins and stuff. And it's really pretty to look at the embryos and watch different things like the actin and other molecules. So it has a short lifespan. It's just a great model organism. I love my worms.
So how did you get into your current project, and what made you choose it?
Yeah, so if anyone's going to Rutgers, you might know that there's an introductory research type class that a lot of freshmen take. It's the honors intro to research in molecular biology and biochemistry with Dr. Vershawn. So I took that my fall semester of freshman year, and then after that I was I should go into research. And at that time I wasn't really sure if I really wanted to go into pre-med. I wanted to weigh out my options. So I was I'm going to get into research. So I started looking at a bunch of labs, and I saw this lab first, and I was I want to go, but my PI is actually in the medical school. And for some reason I was she's not going to take me because I'm just a freshman, and they don't want stupid freshmen or something. So I didn't apply to her first. And I asked a different PI. They said they didn't have space in the lab. So I just asked her, and she was yeah, come on in. And yeah, the rest is history. That was the project they were working on. I got really into it, and now I'm kind of working on it for my senior thesis. I guess I'll get a little bit more into what the science of it is. So when we first started, our PI has published a paper in the past just detailing the guidance receptors and the different molecules that sort of regulate the actin and the different cytoskeletal components during the process of ventral enclosure and other things. So they figured out that SED10, or RAC1 as it's usually called in mammalian systems, is the GTPase that's sort of catalyzing all of these events. So the next step was to figure out what is regulating the GTPase. So for anyone who doesn't know, there are two different types of things that are usually regulating GTPases, which are GEFs, which are guanine exchange factors, and GAPs, GTPase activating proteins. So we started looking into what GEFs could possibly be regulating SED10 in the worms. So at that time, what we were actually doing is there are 19 known GEFs in C. elegans. So we were doing RNAi screens on with all of them. So we were just putting these worms on these RNAis to knock down whatever this GEF might be. And then we would sit there and then we would count the lethality, the embryonic lethality for just said 10 mutants versus the said 10 mutants with these GEFs. And so based on that, we started narrowing down, whatever GEFs we might think it would be. And over time, I guess previous undergraduates have also worked on the GEF that I've been working on, which is SED5, SED12, or DOC-ELMO, as it's called in mammalian systems. And the thing that they found that was interesting is that there was like a sort of dual regulation. It was not doing what they were expecting it to do as a GEF in certain places, but then it was in other places. And so I kind of got sucked into that. And why is it doing two different things? To summarize, I don't know if that was short or not.
When doing background research on you for this episode, I saw that you were credited on tons of different research posters and papers, collaborating with people in different departments and even other universities. So how'd you get involved in so many different projects?
I feel like when you search it up, it probably looks a lot more impressive than it actually is. So some of the posters were probably like with my own lab, and like other students were doing research and they probably just cited some of the images or some of the work that I did in that poster. So that's just one thing. And then last summer, actually, to back up a little bit more, in spring of 2020, I took a class at the Protein Data Bank. It was about molecular mechanisms of drug action in the central nervous system. And so that was super cool. I was always really interested in the Protein Data Bank. And then I found out the class was there. I was like, wow. And then turns out they really liked me and they selected me for an internship. So I was like super, super grateful for that. And the internship, it was a project on COVID-19 evolution. So what we were doing is we were taking the sequences that were being published by like whatever agencies and organizations from like all the COVID-19 testing. And we were taking that and doing like molecular modeling. So I guess some protein structures had already come out of the COVID-19 proteins. And based on that and the mutations we were seeing, we were modeling them to see like how the virus has evolved. And yeah, that was the research of the summer. It was super cool. But yeah, I think that was probably what a lot of those results were. But yeah, it's probably like two things, but it looks more impressive than it actually is.
It's still pretty interesting on its own, though.
Yeah, it is.
I have a question for you with regards to this project, but more in relation to the world situation we were all in at the time. And the topic of the research was related to that. So first question is, I think I understood before, you've been working on your embryonic development lab since freshman year or something along those lines. Were you able to continue your research at the time you were doing your internship? And if so, how did you manage being into different things at once?
To be honest, I didn't really continue it in the summer. I did finish out whatever, like, because I was signed up for research for credit in the spring of 2020. So honestly, I didn't have that much data. So I just like took some movies that people already had and I like reanalyzed them to make sure that we were actually doing the right thing. And then I tried to do like some like protein alignment stuff. And that was like what my paper was then. But in the summer, I didn't really keep up with the lab. I like, there were some meetings that I went to like every now and then, but I wasn't like actively doing research. And even in the fall, I wasn't going to lab. I just went to our like weekly lab meetings, but I wasn't doing too much. It was only in like this spring that I started physically going back to the lab and doing research and getting the credit for it. So yeah, I didn't do two labs at once. I'm not like superwoman or anything like that.
Thank you for your answer. And then my second one is kind of like a follow-up in the sense that when you were working on your COVID research, first of all, how did it feel? I mean, I guess one of our first episodes as a podcast team was reflecting on the difficulties and the challenges that COVID brought to research and undergraduate students. And how was it for you to study the virus that is basically made our generation like so impacted, and like historically has created a new chapter for like future textbooks basically.
Yeah. I mean, it was really surreal because I feel like I'm sure other people felt the same way, but I was having like a really hard time at the time that we went home. And like it was just hard to see like so much death and so much suffering, and to see like the way that our, you know, our presidential administration was handling things, and the selfishness that we saw from people around us. It was really like taking a toll on me. And it just felt like I wasn't being able to do anything to help. Like I wasn't even like going to the hospital to volunteer anymore. Like that was like one thing I was like, oh, maybe we can keep doing that, but then that got shut down. So I felt so useless, and it was just really nice like to have, first of all, just something to like focus my energy on in the summer, and then also to like feel like that something I'm doing might be helpful for some research, even if it's not that much. I feel like I'm at least learning something, and maybe it might be useful to somebody in the future, right?
You've mentioned you're like pre-med. Are you interested in pursuing an MD PhD career, given like your enthusiasm about research? I'm like so intrigued.
Yeah, so that's one that I keep going back and forth on. I think for now I've decided that I don't want to do the PhD. But yeah, last year I was really considering it. I talked to a lot of people to see if that was the right path for me. I think in the end, I don't see myself um, running a lab, to be honest. I feel like I want to practice medicine more than I do run a lab and have to worry about, you know, getting grants and things like that. So I just feel like that is not going to be like a useful investment of my time, even though I may enjoy it. I don't know. I feel like we also have to be a little bit realistic about the way we pursue our education, our career plans. But I think I will still be doing research. I think I'm going to be taking a gap year. So I may do research in that time. I don't know what I'm going to do. But yeah, I'm not doing an MD PhD, but definitely still research while I'm in medical school.
What does your day in a lab look like? And follow up to that, what is the most like annoying or like tedious thing about your lab? Because just to give some people perspective, like lab science isn't always the most glamorous, as you might imagine. So just to get like a realistic picture of what that looks like.
Let's see.
As a
Well,
follow-up, can I ask, how was that changed with COVID, and how do you see it maybe going back to normal or changing again in the upcoming year, which is the last one for all of us actually? So
yeah. So there's always the constant job of having to maintain your worms. Like you have different strains going, so you just have to keep transferring them to different plates like every 3 days. So I usually leave that as like an end of the day job because I'm like, it's not that high priority. If I have any crosses going on or like genetics, I usually start my day off by like looking at where my worms are at, if it's the day for me to continue it on, and if I need to like speed it up by putting it at a higher temperature or something, then that's there. In terms of doing like embryonic lethality counts, you usually have to like pick a bunch of embryos from the plates and make a bunch of pads and count them. I honestly like doing that a lot because it's just like really meditative. I just put on my music or whatever, and then I go sit at the microscope for hours, and I just sit there and count and look at the embryos, which is always fun for me. Same thing with imaging. I really enjoy doing that. In terms of what I don't really like doing, especially when it starts failing, PCR is so irritating. I just hate it so much. Like for a while now, it's been going good. And then I have this one thing that's just not working. And my PI keeps saying like, oh, get new primers, do this, do that. I'm like, I don't want to do it anymore. It's just really tedious, especially when it keeps failing. You're like, oh God, what is the point? So yeah, that is probably my least favorite thing to do. In terms of COVID, when we all went home, like my worms were just like sitting there and I guess they like put them at a lower temperature for a while, but Yeah, my PI was still going in a few times a week and our lab tech was still going in a few times a week, but I honestly didn't have anything to do with it. I just like couldn't worry about it because like what am I gonna do about it? I think yeah, now it's been okay, even though we've had COVID, but we still have masks on all the time, which I don't know. The only annoying thing about that is sometimes it like fogs up the microscope lenses if you're not careful. But yeah, like recently it hasn't really had much of an effect. You're just like wearing a mask all the time, which is not a big problem. But it was more affecting the types of work you could even do when we weren't allowed to go into lab. So that's when I did a lot more of that, like protein alignment and just reading in general, because you just can't do anything else.
Thank you for your answers. Kind of inspired by this research environment talk, I would like to ask you a question that is similar to what we asked right now, but with another twist to it. So we've just talked about what are the things that you like doing or what you don't like doing in the lab, like with regards to your project. But what about research in general? You seem so passionate about it, and I would love to learn what about research exactly makes you happy, or like maybe what are the characteristics of research that make you pursue it as an activity, as you are also a student, and like we've been talking in one of our prior episodes, and at some point today, it is tedious to be both a student and a researcher. And then you mentioned before you used to do volunteering at a hospital. I mean, it must be pretty a hectic lifestyle. So I would love to learn about what about research do you like, and what keeps you going with regards to it.
Yeah, I mean, I think I've always been interested in what research would actually be like. I never really had a clue of what it would be when I was in high school, but I was just like, it would be so cool to work in a lab. And so I started doing it in freshman year of college. And it was really hard at first because you don't know anything. Your skills are not that good in terms of just doing anything. Even the so-called simple things, you just mess it up all the time. It was really hard at first. I'm not even gonna lie. I used to get so discouraged because like simple things would fail all the time. But after a certain point, if you just keep pushing, you'll get better at those things. And then that's when you can start to do the real work. And when you start designing your own experiments and you start like reading up on things and getting ideas on what to do, and then you execute that and you actually get the results you might have expected, or just even something totally new, even if it's not what you expected, it's so cool and it's so interesting because it's like you're the one who's doing it, right? Because no one else has done it before. I mean, we all used to read the stuff in the science textbooks in high school and we're like, oh yeah, I mean, like obviously everyone's always known that, duh. But somebody had to sit there and like slave away to figure out what it was. And it's like, it's super cool because now you're getting to do it. And I've always loved science so much. And I think this is just the embodiment of what I love. And I get to do it myself with my own hands, which is why I'm so passionate about it, even when things get tough.
Has doing research helped you consolidate your passion for medicine and your wish to go into medicine?
Uh, yeah, I think so. I think it definitely has, because I always thought about doing medicine when I was younger. And then I came to college. I wasn't really sure if that was definitely what I wanted to do. So that's why I started doing research. Um, as much as I do enjoy doing the science and just having fun, like being a student, I don't see myself doing it forever. I'm enjoying it right now because I've always loved school. I mean, call me crazy or whatever, but it's just fun to like sit and learn about things. But definitely I know like what I'm missing right now is having that hands-on experience. I was really upset when we had to like stop just doing any sort of like clinical stuff last year. And so that really proved to me, I think I definitely need to go down this path. And as much as I love research, I don't see myself doing just that for the rest of my life.
I think we all felt you when you said you love learning. I think I have a feeling that people that enjoy research and enjoy medicine too, because it is a field that requires you to have to learn so much stuff. we are all passionate learners. So there's no craziness, nothing of that kind associated with it.
Yeah, we're on a podcast where we discuss the stuff we're doing in school, so there's nothing all that weird about it to us.
Yeah.
I have a question about, so at this point, like what are future directions that you're gonna take as a undergrad researcher, and then like for your future? You mentioned you want to go to med school, but have you, I mean, I don't know, I feel like some people know it from the day they were born, and some other people are like, oh, maybe I'll just wait for rotations and I have an idea. But you mentioned you're interested in embryology, and then you did a work on evolution, which to me, evolution a lot of times makes me feel of like progression and birth of something new. So do you have any particular medical discipline that is catching your interest at this point, or do you feel like you don't know yet?
I mean, it kind of goes with it. Like I do have more of a go with the flow attitude about it. I never know. I'm not going to know what happens. I may end up hating what I think I want, but I've been really interested in going down like the OB-GYN path, or even neonatology, which is like super niche. If anyone doesn't know what that is, it's like dealing with premature babies and their development. So that is like a really, really long path if I do decide to go down that way. But that's always been something that's so cool to me. Like I said, I've always been so interested in development. So this lab felt like a perfect fit for me. And I don't know, maybe that's what I'll end up doing in the future.
That sounds so beautiful, and honestly, like so cute. Like imagine babies all day, every day. That's honestly,
yeah,
that's sweet. And like the premature ones also need so much care and attention, and I love it. I wish you the best for it, and I really, really enjoyed them going with the flow attitude. I think it's important in life.
I guess from there, like having that go with the flow attitude, not setting or not being too rigid in what you want your future to look like, and allowing for flexibility and change. What other like pieces of advice or things that you've learned so far in your undergrad journey as a researcher and student and pre-med, can like what advice from that can you give to other people who are maybe in the same situation?
Yeah. I think other people can probably also attest to this, but I haven't been this go with the flow all the time. I mean, I've never been like too strict about, oh, it has to be this way. But if you knew me in high school, I was so, I don't know what was wrong with me. I sat there and I like plotted out what every single course I was going to take in high school, as if that was going to matter at all. Like, I think coming to college, you get a lot more perspective about what your life might actually look like in the real world, even though it's still sort of removed. I think especially in the pre-med world, there tends to be a lot of pushing for just, I don't know, just filling up your schedule like crazy and just not having any time to enjoy yourself at all. And everything has to be a certain way. You can't do anything fun. It doesn't have to be like that. You know, it's open for you. This is your college years. You know, you should enjoy yourself while also studying hard. I think it's important to expose yourself to a lot of different things. I mentioned at the beginning that I'm a religion minor, so that has been super fun for me because I've always also been interested in like religion and how that impacts humanities. So that's like a fun like step away from rigorous molecular biology courses. Yeah, and then I think just keep an open mind. Don't be too strict about things that you don't have to be too strict about, and just be willing to see what comes in the future, and don't like punish yourself for it if it's not what you expected. And yeah, I don't know. I don't really have that much advice. I'm not that much older.
That's fine. I wanted to say like, yes, I knew you in high school, and I was also planning those courses probably with you, trying to see like who's teaching what and why are they bad and why are they not. And then like I've also like watched you like become less and less I guess, neurotic. Well, I was also still neurotic because like we started off going into Rutgers both as CBN majors, and then you were just like, peace out, I'm an MBB major now. And then you got a religion minor, and I'm just like, what is she doing? Why is she doing this? And then like I'm like, oh, okay, she's actually like enjoying herself. So props to you for realizing that before I did. Okay.
Why did you switch? I switched because I would love to learn.
I've always been interested just in the molecular dynamics of things, and that class was super interesting. I don't know, I really wanted to sit and like pick apart like the smallest little details about things. So I just felt like, I don't know, MBB was going to be a better fit. Plus I like chemistry, so I was like, oh, it's perfect, it'll be perfect.
I'm outnumbered here. You're all like all MVP. I'm like the only
Yeah.
one.
I don't know if you guys can relate to this, but I feel so weird when we're like transitioning from a class to a lab environment when they ask you like that serial dilution for real, or like, as you mentioned, like the PCR for real. Like I think we've learned about PCR in at least like 4 classes each, and then you get to the machine and you're like, hello, what language do you speak? I don't know what you are.
I think like the reverse is also like the same. I feel like because I started going to lab in freshman year, and then I started learning, I mean, like my research is focused on like GTPases. And then when I was taking biochemistry and molecular biology, I was like, oh my God, I know about this. Like we're learning about this from a textbook, and I know about this. It's so cool both ways, I feel like.
I really wanted to thank you, Thejasvi, for being here with us today. I mean, it's super, super exciting for all of us every time when we get to, first of all, meet a new student who's like passionate about research. Our intentions with the creation of this podcast is really to get a conversation going about undergraduate research in general, and like learning something new. Like we love learning, we love being exposed to new information, and hearing it from the students themselves. I think it's one of the best ways to learn, like peer-to-peer. So thank you for being here. I wish you, but I'm sure we all do, bestest wishes for the future. Best of luck with your project for senior year. I'm sure with your drive and your passion and your enthusiasm, you will achieve an amazing thesis. And in your gap year, I hope you can acquire all the experience you want and get closer to your dream to practice medicine. And I would like to remind you that you are a great student and you've got a lot of potential. And thank you so much again for tuning with us today.
Oh, of course. This has been so fun. You're so nice. I wish you guys all the best too. And yeah, I mean, this has been so great. It's so fun to talk to people about research who are also passionate about it, because it's hard to find sometimes.
Thanks for not killing me for asking me to be here.
No, I enjoyed it, honestly.
Okay, I'm glad.